Assalamu alaikum, my dear students. So again we will continue with the details of bacterial cell structure, and today we are going to focus first on the cytoplasmic matrix. Cytoplasmic matrix is actually the substance that lies between plasma membrane and the nucleoid. Okay. Uh, One more term that you will remember here. This is protoplast. It is actually the cell membrane and everything within it. Say for example, this is a bacterial cell. Okay, this is cell membrane. Uh, this is nucleoid, and this is the cytoplasm. Everything within the cell membrane and including the cell membrane, this structure is known as protoplast. Protoplast simply means cell membrane plus everything that is within it. The cytoplasmic matrix in bacterial cells, it is largely contained of water. It contains almost 70% water. And under electron micrography, you will see that the cytoplasmic matrix, it is featureless. It does not show any pattern, but you can see it is packed with a large number of ribosomes, nucleic acid you will find, and other inclusion bodies. Okay, so in electron micrographs, under electron microscope, there are no clear-cut patterns, but you can see a large number of ribosomes, you can see nucleic acid and other inclusion bodies. Okay, the cytoplasmic matrix is actually a mixture of carbohydrates, minerals, proteins, nucleic acids, and water in the form of aqueous fluid or matrix. Okay, you can find them dissolved, okay, in dissolved state, and this mixture of carbohydrates, proteins, minerals, nucleic acids, and water, this is known as aqueous fluid or matrix. The cytoplasmic matrix, it lacks eukaryotic features like cytoskeleton. Cytoskeleton is absent; it's not present in cytoplasmic matrix. There is no cytoplasmic streaming, and membrane-bound organelles like endoplasmic reticulum, mitochondria, Golgi apparatus, they are all absent. Okay. Now, first we will talk of ribosomes. These are small bodies made up of proteins and RNA, and they are loosely attached to the plasma membrane of bacterial cells. They impart granular appearance to the cytoplasm. Just जो cytoplasm होता है bacterial cells का, it appears granular because of the presence of a large number of ribosomes inside it. And these ribosomes, they are of 70S type. That means their sedimentation coefficient is 70S. I have already talked about 70S and 80S ribosomes. Okay. In young bacterium, the ribosomes exist in long chains which are held together by mRNA called polyribosomes. अगर आप फास्ट डिवाइडिंग बैक्टीरिया को देखोगे, उसका सेल स्ट्रक्चर देखोगे, okay, you will find large number of ribosomes. They are attached to mRNA in the form of chains, and this whole structure is known as polyribosome. Many ribosomes are attached to the mRNA. Then cytoplasmic inclusions. Okay, cytoplasmic inclusions are actually the organic and inorganic storage granules which are visible under light microscope. Under inorganic inclusions, we have volatile granules which contain phosphate, sulfur granules containing sulfur, then magnetosome, they contain magnetite. Okay. Organic inclusions include glycogen, which is a carbon reservoir, polyhydroxybutyrate, carbon reservoir, and then we have gas vacuoles. Gas vacuoles are actually the organelles. They are the structures which provide buoyancy, which provide support, which provide balance to the bacterial cells, and they are mostly present in blue, green, purple, and green photosynthetic bacteria. Now the nuclear material. You know that the nuclear material of bacterial cells, it is not delimited from the cytoplasm by nuclear membrane. Okay, you can simply say that the bacterial cells are prokaryotic. They possess no well-defined nucleus and nuclear envelope. Okay, the genetic material in bacterial cells, it is single circle of double-stranded DNA, and this single circle of double-stranded DNA, it is divided of histones, which are known as basic proteins. Okay, and this single circle of double-stranded DNA, it is also known as bacterial chromosome. Okay, this bacterial chromosome or genetic material, it is located as a diffuse, irregularly shaped material in the cytoplasm called nucleoid. That region of the cytoplasm is known as nucleoid. Nuclear body, nuclear area, chromatin body, or genophore. Okay, nucleoid is amorphous lobular mass of amorphous mass of fibrous, intensely chromatic material occupying about 10 to 20% of cell volume. So what is nucleoid? Nucleoid is actually the region of the cell which contains the nuclear material. Okay, it forms about 10 to 20% of the cell volume. This volume of cell is known as nucleoid. Then this nucleoid is composed of about 60% DNA. The region which is known as nucleoid, it is composed of this region. It is composed of 60% DNA, 30% RNA, and 10% protein. And these 10% proteins, they mostly represent RNA polymerase. Okay, they mostly represent RNA polymerase. I will repeat once again. That region of the bacterial cell which contains the nuclear material, okay, this is known as nucleoid, and this nucleoid forms 10 to 20% of cell volume. Nucleoid is actually composed of about 60% DNA, 30% RNA, and 10% RNA polymerase. This RNA polymerase is an enzyme which is involved in the formation of RNA from DNA. Okay. Now plasmids. Plasmids are basically small circular extrachromosomal double-stranded DNA molecules. They are small circular extrachromosomal. That means this is the bacterial cell. It contains here nucleoid. Okay. In addition to nucleoid, you will have some small circular extrachromosomal. That means they are independent from the bacterial chromosome. Double-stranded DNA molecules. They are the double-stranded DNA molecules, and these exist and replicate independently of the bacterial chromosome. I have already told you they are not associated with the bacterial chromosome. Whatever, their replication is independent. Okay, their existence is independent. Or in some cases, you will find that these plasmids they integrate with the bacterial chromosome. कुछ cases में ऐसा देखा गया है जो bacterial plasmids है, they are associated with the bacterial chromosome. These plasmids, they are inherited to the progeny. That means when bacterial cell divides, this is nucleoid, this is a plasmid. When bacterial cell divides into two cells, each cell will receive a copy of this plasmid. Okay, ये भी divide हो जाएगा. Okay, it will replicate and will transfer to both the daughter cells. The plasmids are not involved in growth and viability of bacteria. They have nothing to do with the viability and growth of the bacterial cells, but they are known to carry some non-essential genes. Okay, and these non-essential genes means those genes which are not involved in the growth and survival of the bacteria. Instead, they confer some additional properties. Okay, and these additional properties include fertility. They determine whether the bacterial cell will be male or not. Okay, जिसको हम बोलते हैं मेल फैक्टर और एफ फैक्टर. ओके, देखो एक बैक्टीरियल सेल है, if it contains this F factor which is a plasmid, okay, it will be treated as male, and if it does not contain that plasmid, it will be treated as female. Okay, then resistance to antibiotics. Then we have another type of plasmid that is known as R plasmid. The bacterial cell that possesses this R plasmid or R factor, it will be resistant to antibiotics. Okay, मैंने आपको पहले ही बोला है, they confer additional properties. They do not increase the growth and viability of bacteria. Instead, they confer some additional properties. Those bacterial cells that possess the F factor, they will be treated as male. They determine the maleness of the bacterial cell. Those plasmids which are known as R factors, if they are present, in the bacterial cell, these bacterial cells will be resistant to antibiotics. And other plasmids, they determine the pathogenicity. They determine whether the bacterial cell will be pathogenic or not. And some plasmids determine whether the bacterial cell will be able to show the new metabolic activities. Say for example, this is a bacterial cell, it possesses this plasmid, and because of the presence of this plasmid, it has some unique metabolic activities, and these unique metabolic activities are not present in other cells. Okay, this is all for today. See you soon with some other details on the bacterial structure. Till then, stay blessed and thank you.